energy being taken into the cell. And so these are this figure describes a cell that's like our cells. It's a chemoorganoheterotroph, taking in some kind of carbohydrate, in this case glucose, and extracting energy out of that material. And you can see the energy being extracted out. And like I said last time, I'm not sure why they show the energy being dumped outside of the cell. Usually the energy is going to be used by the cell. But the CO2 would be a waste product that would be dumped outside of the cell. The cell has to synthesize other complex carbohydrates for its own purposes. That may happen, and sometimes those are going to be produced by the cell. Nucleic acids are going to be produced, amino acids may be produced, lipids will be produced, and again, those are all those macromolecules that we talked about in Chapter 2 that are needed for cell growth, and the cell's got to be able to produce those. And again, most of the carbon comes from carbon sources outside of the cell when we're talking about chemoorganoheterotrophs. And so you remember from anatomy and physiology, and we've talked about this a little bit already, that the glycolysis pathway, or glycolytic pathway, is one of the 3 central metabolic pathways. And glycolysis takes glucose, which is a C6 molecule, 6 carbons, and it breaks it into 2 pyruvates. And you already know that from anatomy and physiology, and probably from some other classes too. And glycolysis produces a little bit of ATP. What do you remember? How much ATP does glycolysis produce? It produces 4, but it takes 2 to get started, so the net gain is actually 2. Good. All right, it produces some reducing power. And when you hear reducing power, students always ask me, what does reducing power mean? What do you know about reducing power that's being produced out of glycolysis? What kind of chemical is that? You got it, NADH, good. NADH is produced in glycolysis, and get a couple of NADH out of glycolysis. And then we get a lot of precursors, and sometimes those in the text are called precursor metabolites. What do you know about precursor metabolites? What are they used for? The next step, and the next step, what kind of metabolism are we talking about here when we're talking about glycolysis? Remember, there's 2 types of metabolism. We're talking about catabolism, good. And so these precursor metabolites, where do you think they're going to be used? They're going to be used in the anabolic pathways to build up things. So the precursors are the molecules that are needed to make those bigger molecules in anabolism. If you're making proteins, the precursor metabolites are going to be the amino acids that are needed to string together for those proteins. If you're making lipids, then it's going to be the 2 carbon molecules that are stuck together to make the fatty acid chains of the lipids, or the glycerol that's needed for the backbone of those fatty acids. And so those are all the precursors, and those guys come out of, a lot of them come out of glycolysis. Then there's the pentose phosphate pathway, and you guys kind of drew a blank, if I remember right, when I asked you about the pentose phosphate pathway from anatomy and physiology. But a couple things happen there. We get this biosynthetic electron carrier that's produced there. And then we get something else important out of the pentose phosphate pathway. And I told you what it was last time. I want to see if you remember. But it's another precursor metabolite that comes out. Do you remember what that is?
Just from what?
From pentose phosphate pathway, a precursor metabolite that comes out of the pentose phosphate pathway. What was it? Pyruvate comes out of glycolysis. I mentioned it too. If you're a growing cell, you need to make some really long macromolecules. And the backbone of those really long macromolecules has this precursor. The ribose comes out of the pentose phosphate pathway. Just remember, ribose is a 5-carbon molecule. That's how this pathway gets its name, because pentose means that there's a carbohydrate that has 5 carbons in it. And so that's how the pathway itself gets its name, because that's where the ribose for... What kind of molecules would have ribose in them? DNA, RNA, which part of the molecule? the backbone, right? That ribose or deoxyribose phosphate backbone of the DNA. If it's DNA, it's going to be deoxyribose. If it's RNA, it's going to be ribose phosphate backbone. So that all comes out of there. There's another molecule, a precursor metabolite that comes out. It's erythros 4-phosphate, and that one's used to make a couple of different amino acids, including histidine. All right, then there's the tricarboxylic acid cycle. Do you guys know that as a couple of other names? What are the other names for the TCA cycle? Krebs cycle, and I heard the other one? Citric acid cycle. Do you remember why it's called the citric acid cycle? The first product that's produced in this cycle is citrate or citric acid, and that's why it gets that particular name. So the citric acid cycle takes the pyruvate that comes from glycolysis and further oxidizes it, and out of that process you get some more reducing power. Last time we talked about two types of reducing power that come out of the TCA cycle. What are those two types of reducing power that come out of the TCA cycle? The first one was not correct. NADH is the one that can reduce because it's the one that carries the electrons. And then what was the other one? FADH2. So if I ask you where the reducing power is, make sure you tell me the particular version that's actually the reduced form, the one that has the electrons on it. Otherwise, the oxidized form can't reduce much of anything. All right, and then we get some precursor metabolites out, and you know that we get some ATP out of that process. The ATP from the TCA cycle and the ATP from glycolysis are made through what process? We mentioned that last time too. A couple different ways. We talked about 3 different ways to make ATP last time. Which of those 3 methods actually is used to produce the ATP in both glycolysis and the TCA cycle? Well, it's got a specific name. Yeah, it's a type of phosphorylation. And what type of phosphorylation is it? We talked about 3 types last time. Oxidative? Not oxidative. Got rid of one of them. Now we only have 2 left. Of the other 2 that are left, which one is it? Substrate level phosphorylation. All right, so we used the term substrate last time. I want to make sure you have that in your active vocabulary. Do you remember what a substrate is? It's the starting compound that feeds into a reaction. So I was going to do it on the whiteboard. I think I'll just do it right here. And if we were to have this particular reaction, and we have a substrate, and it gets reacted upon, and then we get product 1 and maybe product 2 out of that reaction. This starting compound is the substrate, right? And in both glycolysis and in the TCA cycle, we have some molecules that look something like this, and there's going to be a phosphate on those molecules that's attached by a high energy bond someplace on that molecule. And that particular molecule, or those particular molecules, can serve as this substrate for this particular reaction. And if one of the reactants in this reaction was ADP, then this phosphate group right here can be added onto ADP to actually make ATP. And that's substrate level phosphorylation. Everybody good with that? Because the substrate that's right here, this starting compound, is carrying this phosphate, and this phosphate is ultimately added onto ADP to produce ATP. Everybody good with that? So that's called substrate level phosphorylation. We'll look at it again in a minute, but you see it happening a couple places in glycolysis. It happens at the bottom of the TCA cycle as well. And so all of those occur by this same process. Some substrate, not necessarily this exact substrate that I drew right here, but some organic substrate that's carrying a phosphate attached to it with a high energy phosphate bond is able to transfer that phosphate to ADP and produce ATP. That's what substrate level phosphorylation is. Everybody cool with that? Okay, cool. Let's see if I can get back out of here now. All right. So we get those things out of glycolysis, and let me just go back to the previous slide, and pentose phosphate pathway and the tricarboxylic acid cycle. We're going to talk about each one of those in a good bit more depth, because I really want you to understand these pathways much more than you did when you came out of A&P. They're very important to you. If you remember E. coli, you can feed it glucose and it can make everything it needs. It can make every one of the 20 essential amino acids. It can make all the lipids it needs. It can make all the carbohydrates it needs, all the ribose it needs, all the bases it needs for its nucleic acids. It can make everything from those. And so you need to be able to understand, and many organisms can do exactly that, and you need to understand how they do that, because many of these molecules you end up eating in those vitamin supplements and other things that are actually produced by microorganisms. So I want you to understand how they do that. So those three pathways we call the central metabolic pathways, and they include glycolysis, pentose phosphate pathway. Sometimes we just call it the PPP or the tricarboxylic acid or TCA cycle. You knew that. And again, the key outcomes are production of ATP. That's the energy currency of the cell. That's where a good bit of it comes from, out of all of it comes out of those pathways. We get some reducing power, which actually produces some, not all of it comes out of those pathways. A good bit comes out of the electron transport chain, right? Only a little bit comes out of these pathways. But the reducing power then can lead into the electron transport chain and make more ATP there. And then we get precursor metabolites that are those building blocks for making those macromolecules. And so the book uses figures that are like this, and all the way through you're going to see these little things that look kind of like a flash of light. Those are reducing power, and the reason they look like flashes of light, those are electron carriers. They're carrying electrons. They used to have them look more like lightning bolts, but you can kind of get that idea from looking at those little flashes there. Those are all places that reducing power comes out of those three pathways. And what are the molecules that have reducing power that come out of these pathways? Name them for me again. There's three.
NADH.
NADH.
FADH2.
FADH2. And the other one you're not so familiar with because it's not used in the electron transport chain, but it's used to power a lot of enzymes for biosynthesis. NADPH. Good. NADH, FADH2, and NADPH. Okay? And those come out of these various places. And you can see also in this schematic figure, they show you where the ATP comes out. Along the way, we get some out of glycolysis. Keep in mind, those arrows where they're saying they come out are not exactly precise because the ATP doesn't actually come out of glycolysis until the very last step of glycolysis, the ATP that you build there. And then you get a little bit of ATP out of the TCA cycle. It actually comes down from right about the bottom of that TCA cycle. And then in respiration, we can get a lot of ATP produced that way. Okay, good. So that's an overview of catabolism. Basically, we call it respiration, and respiration is the electron transport chain. And my brain needs to wake up a little bit this morning. And so respiration transfers electrons from glucose into the electron transport chain. That's the process. Moving those electrons that are in the glucose molecule into carriers that can move it into the electron transport chain. And those electrons then basically provide the energy that drives those systems. Just like the electrons flowing through these fluorescent lights are providing the energy that makes that chemical fluoresce, the electrons that feed into the electron transport chain provide the energy to drive the processes in that electron transport chain. And so the electron transport chain, one of the things that it does is it generates this proton motive force. Remind me what a proton motive force is. What does that mean? What are protons? Small positively charged molecules, we call them hydrogen. And then what does motive mean? Something that's going to move or has the potential to move. And force, means power, right? The energy. So now we've got protons that want to move, and when they move, they have power to do something else. And the power that they have to do is to synthesize ATP. In a little bit, I'm going to show you that they have power to do much more than that. And we'll talk about how that works in just a little bit. So we get these electrons. They basically feed into the electron transport chain, and now we get ATP being produced, and this time it's produced by a second method that was mentioned earlier incorrectly. This time it's oxidative phosphorylation. Oxidative phosphorylation happens at the electron transport chain. Okay? And so we'll talk about that. In the case of aerobic respiration, the terminal electron acceptor, you should remember that when those NADH or FADH2 carry the electrons to the electron transport chain, those electrons are very high energy electrons. They've got lots of energy, and they're released from the NADH and the FADH2, and the energy is used to drive those enzymes that actually are proton pumps. Those enzymes that use the energy are proton pumps, meaning they can grab protons from the cytoplasm and pump them outside of that membrane. That process of pumping the protons takes energy, and the energy comes from the electrons that were carried over there by the NADH or the FADH2. Everybody good with that process, right? So each time you pump electrons, some of the energy is pulled out of those, or each time you pump protons, some of the energy is pulled out of the electrons to pump those protons across the membrane. Okay? Finally, when the electron gets to the end of the process, and it goes through a number of chemicals, and each time it's losing a little bit of energy as it's being passed along, finally you've got a low energy electron at the end of that process of the electron transport chain. And that low energy electron has to go someplace. And if there's oxygen around it, then the terminal electron acceptor, the acceptor that's going to take that low energy oxygen is going to be, or low energy electron is going to be oxygen. Okay, so oxygen will absorb that low energy electron, and oxygen will get a little bit of a negative charge. You know that from previous chapters, in chapter 2, we talked about how oxygen likes to grab electrons. It doesn't share them equally when it's in other molecules. It tends to grab them more, and in this case, now we've got an electron. It gave up a good bit of its energy as it was flowing through and charging those various systems, but it's still an electron, and it has to go someplace. If